Okay, so we are done with the muscle proteins. So you should know that there are three groups: contractile, that you have myosin and actin; regulatory, that you have troponin and tropomyosin; and the third one that is the structural, that here we only pick titin and dystrophin. But there are many more. And here we finish this section. Now we're going to study this picture of the sarcomere. Okay, so we're going to dissect all these sections and try to understand why you have parts that are more darker and other ones that are lighter. So here you have two schematics of your textbook. Here you have a myofibril on top, and here in the bottom you have the sarcomere with the detail of what happened between the two Z disc. Yeah, from here to here. So here we are just zooming in into the dynamics, zooming into what is happening in the myofibril. Okay, so we're going to talk about the following component: the Z disc, A band, I band, H zone, and M line. Okay, so let's go on and start talking of each one of those. So let's start talking about the first component. Here is the Z disk, and it's not only located in one in the left extreme, it's also located here in the right. And if you see, they both are darker regions that are actually, if you see the schematic, let's go and see the schematic, you find that they are proteins, alpha actinin, that are attaching to the thin filament of actin, and they separate one sarcomere from the next. So remember the Z disc, they are narrow regions that have a plated shaped regions of dense material, and this is why they look darker. And the second thing that you need to remember is that they separate one sarcomere from the next. Now we're going to talk about the band that is in the middle, that is the A band. And if you see, it looks dark, yeah, darker than most of the other regions. However, if you see, it has like a gradient of colors here, here, and here. So you can clearly identify one, two, and the third that it will be the M line. Okay, and to understand why you find all these gradients, so you find this dark area in the A band or darker than the other bands. So what is happening there? To understand, we need to go to the schematic and you just identify here the A band. And in the A band that you can see here, you will see the three regions. So the region number one is the region that there is a zone of overlap between the myosin in red and the actin here in yellow. So they are overlapping there. So why this section of the A band it looks darker? Because both thick and thin filaments are overlapping there. Then you have the second region here, yeah, that you can only see the thick filaments, the filament of myosin, and you see uh, white regions and dark regions here. Yeah, if you remember, white regions and dark regions is because here you're missing the other, the thin filaments. And finally, here you have the M line that is give stability to the sarcomere, and it's another type of protein. Okay, so this is why you have these three colors in this dark A band. So things to remember: remember the A band is a dark middle part of the sarcomere, and it extends the entire length of the thick filaments, that in this case is the myosin that you see there in red, and include a part of the filament of actin, not all of them. Here, for example, you see that in both sides you see that this in the I band you find also filament of actin, but that those filaments of actin overlap with thick filaments of myosin. Okay, so now let's go to the next section of the sarcomere. So let's move into the I band. And here in the I band, you see that there are lighter regions that I'm going to put a one here, one here, one here, and also you have also a dark region here that is the Z disk. Okay, so you have these two. And I wonder, you think, why do you think that you see these lighter regions, yeah? And why? So to answer that question, you need to see this schematic, and I'm sure that you already did, and you see that it's very clear. Well, the dense area before you have all these filaments overlapping, here you don't have thick filaments, you don't see any myosin. What you see is only actin and titin here attaching, and of course the Z disc that we were talked before is here, that is creating structural stability in the Z disc. Uh, and that is from why this protein alpha actinin. Okay, so that is uniting all of this. So this is why it looks darker. Okay, so remember the I band is lighter because it's less dense area of the sarcomere, because the proteins that contains are only thin filaments, but not thick filaments. Okay, so no myosin in that area. And the third thing that you find a darker region in the middle, that is the Z disc, that pass through the center of each I band. Okay, and to finish, let's move into the next section. And I already talked about a little bit about this H band, and this H band is a narrow region in the center of each A band. So here you see the A band that contain also this region, and in the middle you have this H band. And so it's very clear what is happening when you look at this schematic. We don't have the overlapping zone. This is not here. This overlapping zone is not here. Yeah, so you only have a region that have the thick filaments. So the H zone, as we said before, is a narrow region in the center of each A band, and contain thick filaments but not thin filaments. To finish, I just wanted to show you this electron microscopy picture of the skeletal muscle, and here you see our friend the sarcomere again. Here you see one, two, there are more, three, before there was another one, four, and you can count, and there are so many in this small region. And if you remember, these are just protein thin and thin filaments forming all these bands and zones that we just talked. So this is 3000 magnification, and the magnification is so high that you can see other structures too, like the mitochondria. And in the next section, we will talk about the importance of the mitochondria, and more than the mitochondria, of the ATP. Yeah, the ATP that is very important for the muscle contraction. And as we also talked before, the importance of calcium. Yeah, these molecules are very important for this mechanism of the sliding of these filaments. Here we are seeing in this picture in the electron microscope picture this still, but actually what is happening is that it's constantly under contraction, and to do that you need the other organelles. This is why you have so many mitochondria and the sarcoplasmic reticulum giving ATP and calcium to move between stages of contraction and relaxation. So let's continue. So this is going great. We just talked about muscle proteins, and you already know that you have three types of muscle proteins: contractile, regulatory, and structural. You also now know all these different components of the sarcomere, all these discs and bands, and more important, you know why you see dark areas and light areas. Now we're going to put all this together and explain this sliding filament. I'm going to have a link of a video of your textbook that is great, that I think it summarizes everything and help you to understand things in much more detail. And I'm going to just quickly emphasize that there are 4 steps that they talk in your textbook. And I'm going to go very quick because you already watched the video and I think it summarizes all the information. So we are going to start talking about the sliding filament mechanism, and here we just were talking about all the section before about this, the pictures and the schematics. But as I said before, rather than a static picture, what we're going to have is a dynamic process. So here in the beginning, you see this is a relaxed muscle. Yeah. Here you have a partial contracted muscle, as you can see in this picture, and you can also have a fully contracted muscle in this picture. And the picture that we study in the beginning, this one is going to be modified. Yeah. So here you see that the actin is going to start getting closer and closer together to the M line. And the same thing happen is going to in the schematic. So the actin is going to be pulled together here. So what you're going to end up is with a sarcomere that is much shorter. Yeah, compare this is much shorter than the other one. And this is an example of one sarcomere. This muscle to be contracted require that all the muscles in the fiber contract. Okay, so just to recap, I told you that there is something called the sliding filament theory. And according to that, muscle fiber contract when the myosin pull the actin filament closer together. In addition, I told you that this lead that the sarcomeres become shorter within a fiber. And finally, when all sarcomeres in a muscle fiber shorten, you have the fiber contract. I want that you have 3 main ideas from the sliding filament mechanism. First one is that here this is a representation of your friend myosin, and what is myosin doing is pulling the actin, and thus is causing that the thin filament to slide inward. So it's sliding here in the beginning a little bit. Here you see that it keeps sliding, and here it keeps sliding. And not only there, the other region also is sliding and sliding and sliding. And as a consequence, you can see that there is a complete overlap. Second thing that you need to remember, Z disk. The Z disk start far from each other, then they start getting closer, and when there is full contraction, they are much closer to each other. In other words, as a result of the pulling, the Z disks move toward each other, the sarcomere shorter from a initial stage when they were very far when the muscle was relaxed to be very close together or much closer together when the muscle is fully contracted. The third thing that I want that you remember from the sliding filament mechanism is that in addition to actin and myosin, you have all these structural proteins. And for example, titin is there, allowing that the muscle contract in a uniform way. Yeah, so thanks to these structural proteins, there is a transmission of force to the entire muscle, resulting in a whole muscle contraction. Now, I'm going to show you a video. I'm also going to show the video or put a link for the video of the Wiley textbook that I couldn't add. But here there's one that I found that it was also pretty good that explained everything and summarized all this sliding mechanism and allow you to see what is happening in a lot of detail at the protein level.
Muscle contraction is at the basis of all skeletal movements. Skeletal muscles are composed of muscle fibers, which in turn are made of repetitive functional units called sarcomeres. Each sarcomere contains many parallel overlapping thin, actin, and thick myosin filaments. The muscle contracts when these filaments slide past each other, resulting in a shortening of the sarcomere and thus the muscle. This is known as the sliding filament theory. Cross-bridge cycling forms the molecular basis for this sliding movement. Muscle contraction is initiated when muscle fibers are stimulated by a nerve impulse and calcium ions are released. The troponin units on the actin myofilaments are bound by calcium ions. The binding displaces tropomyosin along the myofilaments, which in turn exposes the myosin binding sites. At this stage, the head of each myosin unit is bound to an ADP and a phosphate molecule remaining from the previous muscular contraction. The myosin heads release these phosphates and bind to the actin myofilaments via the newly exposed myosin binding sites. The two myofilaments glide past one another, propelled by a head-first movement of the myosin units, powered by the chemical energy stored in their heads. As the units move, they release the ADP molecules bound to their heads. The gliding motion is halted when ATP molecules bind to the myosin heads, thus severing the bonds between myosin and actin. The ATP molecules are now decomposed into ADP and phosphate, with the energy released by this reaction stored in the myosin heads, ready to be used in the next cycle of movement. The myosin heads resume their starting positions along the actin myofilament and can now begin a new sequence of actin binding. The presence of further calcium ions will trigger a new cycle.
I think that video is great and it helped you to summarize all the information. Now I'm going to go and talk about the 4 steps. Yeah, the key components of the video that you need to remember for your test, that your textbook divided into 4 main sections. Let's start talking about the first 2 steps of the contraction cycle. And the first step is the ATP hydrolysis. In the ATP hydrolysis, you have ADP, phosphate, and energy. So, in other words, the myosin to move and attach to the actin, and here this is why you have an angle. So it's oriented to the right in the right way to attach there. You need to hydrolyze the ATP. This is why it has a binding site, and here you see the ADP and the phosphate. So remember, first step, the myosin head hydrolyzes the ATP and becomes energized and oriented. The second step, you can see it here, is the cross-bridge attachment. So you remember that here they were getting close, yeah? The actin and the myosin, they were getting close, so now they form a cross bridge. And so here the myosin heads binds to the actin and form a cross bridge. Now we're going to talk about the third step that is called power stroke, and this is the key force generating step. And here the summary of everything is here. The myosin pull the actin, and as a result of that, the myosin protein reverts back into its original conformation. There are 3 small steps. First step, phosphate is released. Then you find that the myosin filaments move the actin toward the center of the sarcomere, and then here the ADP will be also released. And so here the key thing that you need to remember is the importance of the myosin head to pivots and to pull the thin filament past the thick filament toward the center of the sarcomere, and this is what you call the power stroke. Okay, so let's move to the last step, that is the step number 4. The last step, the step number 4, is called cross-bridge detachment. In other words, the actin and myosin here, one is going to go here, the other one is going to go here, they are going to separate, and the first thing that happens is that the ADP molecule, as we talked before, is not has a low affinity here anymore, so it's removed from there and there is now a space. So a new molecule of ATP will be there and is going to attach to the myosin. Yeah, so now that you have a molecule attached there, the the process is already resetted and the ATP can be again hydrolyzed to start a new muscle contraction and shortening of the sarcomere. The cycle can continue as long as there is a nerve impulse, calcium, and ATP available. So to summarize here, what happened again, the myosin head plays a key role because it binds again to the ATP, resetting the process. And as the myosin has a higher affinity and is there, the cross be detached from the actin. We finished the section number 3, and at this point you should know all the muscle proteins. You should know that there are 3 types: contractile, regulatory, and structural. You should also know that there is a structure called sarcomere, that they have different components, that the sarcomere is the unit of contraction of the muscle, and at the end we watch a video and I gave you some information about, or summarize the 4 key points about the sliding filament mechanism. And we are done with that. So if you have any questions, just let me know and you can send me an email.